Hello everyone, I'm Shayan Tabe Bordbar from University of Illinois at Urbana-Champaign, and this is thermodynamic-based modeling of enhancer RNA transcription in breast cancer cells. Estrogen receptor is an important transcription factor in the context of breast cancer. We aim to study the transcriptional program mediated by estrogen receptor in an integrative framework. We use enhancer RNA transcription as an indicator for activity of enhancers and use standard classification methods as well as thermodynamic-based models to answer the following question. Why some regions with active enhancer mark are transcribed upon treatment with estrogen while others are not? Answering this question will also learn important factors responsible for the ER-alpha mediated transcriptional program. Here is an overview of our pipeline. We define a binary classification task where positive class of enhancers are the ones harboring enhancer RNA as well as ER-alpha chip peaks and H3K27AC mark, while the negative class has ER-alpha and H3K27AC marks, but it lacks enhancer RNA transcription. We use motif features to build an extensive feature set using motif strength and pairwise adjacency of the motifs. This large set of features is then fed to a random forest model in order to extract the top most distinguishing features. The features in terms of transcription factors and interactions are then used in a thermodynamic-based model to gain insights into ER-alpha regulatory network in order to be able to point out the mechanisms underlying a variance impact. Here we describe the results of random forest. Panel A is a Venn diagram that illustrates the composition of each class of enhancers, while panels B and C show several features based on motif scores that are significantly different between the two classes of enhancers. Full set of features was fed to a random forest model, and the most distinguishing ones were extracted to be incorporated in a thermodynamic model. You can see that the trained random forest model is able to distinguish positive and negative classes of enhancer on held out tests. GEMSTAT is our thermodynamic model of choice. An ensemble of 4,000 GEMSTAT models were constructed based on the extracted features and trained to solve the defined classification problem. Top performing models on a validation set were then chosen to be considered for the rest of the study. The performance of these models on a held-out test set is comparable to that of random forest. Here we describe the results of an in silico knockdown experiment where we removed TFs or TF-TF interactions one by one and looked at the results. Each pair of bars represents a transcription factor or an interaction. The top panel shows the percent of the enhancers significantly affected by the removal of that feature, and the bottom panel shows the direction and the extent of the effect on the enhancers. The roles learned in this way are in line with literature results. For example, FOXA1 is a key determinant of estrogen receptor function and endocrine response, and this is what we learned using our models. NKX31 and LEF1, known as repressors, are found by our models. NR5A2 is an activator, correctly learned by our models, and YBX1 is a repressor, correctly learned by our models. Next, we quantify the impact of sequence variance, including SNPs and short variance, on the enhancer activity. Panels A, B, and C show how our model was able to prioritize breast cancer or tissue-related variants better than a location-based or random method, while panel D shows that the predicted and observed target enhancers of an AP2 knockdown experiment significantly overlap, where the observations are obtained in an AP2 gamma knockdown Grosick experiment. Finally, we look at three interesting variants more closely and show potential mechanisms underlying each of these variants. In panel A, we propose that RS380926 creates a potential FOXA1 site in the promoter of CHPT1 gene, leading to its increased expression in breast cancer. In panel B, we speculate that deletion of a REL-A or NF-kappa B site decreases the activity of an enhancer associated with NOX-RED1 gene. And in panel C, we show that the somatic variant causes decreased predicted binding for NR5A2 transcription factor, and hence decreased activity of this enhancer, which is related to KEAP1 gene, a key tumor suppressor in breast cancer. In conclusion, this is the first thermodynamic-based model applied to mammalian system that uses enhancer RNA transcription as an indicator for enhancer activity. This modeling enables us to perform mechanism-aware variant impact prediction, and the results are in line with literature evidence. Thank you very much for your attention.